Good morning and good afternoon and good evening. And today is the second International Whole Brain Architecture Workshop. And organizer is Whole Brain Architecture Initiative supported by Transformative Research Areas, Biology of Behavior Change. Thank you for your participation. This is a general programs today. For the first welcome and opening remarks is provided by Hiroshi Yamakawa, WBAI. And then a guide for audience, I will introduce how to listen to what is the VRA data or something. And next 9 to 20 to 10:25 oral presentation, 3 presentation is provided, then break. And from 10:35, a panel discussion will be held. Okay, so please, for the first time, please note the event guidelines, so no screen recording. And if you have any questions, please submit via the chat, and selected questions addressed during the Q&A. Okay. So for the first introduction, so welcome to the second WBA workshop is provided from Hiroshi Yamakawa. Please, thank you.
Thank you to the introduction of Tawatsuki-san. I'm very honored to organizing this second WBA workshop in the brain behavioral change hackathons. So this is the second workshop, and there are some speakers and panel talk. And today the leader of this behavioral change area is Masaki-san is coming, and thank you for coming here. So we will start the second W workshop. And this is the very short introduction of the whole brain architecture approach. Whole brain architecture is almost started 10 years ago, and to make the brain inspired AGI, Artificial General Intelligence, mimicking the brain architecture. That's about thousands of software component is organized like a brain. And this idea is maybe some benefits for that highly compatible with the humans, so that they will have the brain-inspired interpretability and mediated with the super intelligence. So today, this kind of AI safety is a very important issue because today's artificial intelligence is progressing very fastly. As you know, that there are language model. So, but today's language model is a machine learning-based model, so we cannot understand the insights of these systems. But we can build the AI on the AGI like a human brain. We can understand the more in the inside of their thinking and their emotions, something that's kind of many aspects of the computation. So it is important to think about the brain-inspired AI. Now we are thinking about some idea. The BRA data is approach is making the data like a brain. So a brain reference architecture, and that interpretability is a basis of the understanding the brain like AI. So we can control and monitoring these AI, and that kind of nicely monitored and controlled AI can be used as a mediate the communication of the other AI, not machine intelligence AI and human, and there are something bridging these communities. And other aspect is the brain-like AI has maybe the thinking about everything like a human. So the community of the brain-like AI can be more human-like culture is developed in that kind of community. So there are many good things of the human-like AI. So now we are progressing these kind of areas. So and This is a short story of the WBA approach conducted by WBAI. This is established in 2015, and after that, we are decided to promoting the development of the WBA approach. And at that time, after that, 2017, we have defined our mission and vision, so to create a world with AI existing in harmony with humanity. And after that, about 2020, we have established a method for building the brain-like AI that is called the BRA-driven development. Then now we are doing this kind of activity, and we'd like to. This method is almost something like this. And BRA-driven development is based on the neuroscience paper and data, and we make the anatomical structure as a brain information flow. And after that, we make the hypothetical component diagram. That is kind of the specification of the software. And after that, we build the brain-inspired software. This three-step design method is very useful to make the brain-inspired software, because the upper side of the area can be conducted by the engineer, and the lower side is mainly conducted by the neuroscientist. So the expertise of the people can be separated. So this makes collaboration is efficiently. So just this is related to publication, and so our future target is making the brain-inspired AGI, and now we are almost finishing the brain-inspired data, and until the 2017, 2027, about that. 2 years later, we'd like to make the first version of the all brains BRA data. So after that, we can progress all the full brain software. We call that whole WBRA is the whole brain reference architecture until the 2027. And today we are this is the progress of the our activity is making the BRA data, and that is stored in the brain reference architecture editor system, and you can see the data in here, and you can access. So this workshop's aim is promotion of interaction or collaborative creation of the BRA content, and promotion of interaction, internationalization of the BRA content. So thank you for hearing. So please go to the next step.
Thank you for introduction. So we would like to move on to guide for audience. So I would like to share what is BRA data, and because it is very difficult and complicated thing. So first of all, I would like to introduce what the BRA data is. Okay. So I'm Yoshimasa Tatsuji from the Whole Brain Architecture Initiative. Then today I'd like to talk about BRA data. So out of my talk is like this. Sorry. Okay, my talks are about this. So first of all, I would like to give a brief explanation of what the brain reference architecture data, shortly BRA data is. As already explained, BRA serves as a reference architecture for software that mimics cognitive and behavioral function in a brain-like manner. BRA consists of BIF. BIF means which is information about neural circuits, and HCD, FRZ, which is information about functions realized by these neural circuits. Briefly speaking, BRA is a kind of design specification of brain-morphic architecture. BIF serves the foundation of the architecture, and HCD and FRZ represent what and how the architecture realizes the functionalities. So here I will briefly explain the design methodology of BLA data known as the Structure Constrained Inference Decomposition method, shortly SCID method. First step is brain information flow BIF construction. BIF is constructed based on the neuroscientific papers and data about the anatomical structure, where the uniform circuits and their connectivities form neural networks. Uniform circuit is a very important concept. It is a group of neurons composed of specific cell types within a specific brain region. Importantly, uniform circuit is a functional unit which codes for sole functional meaning. And then aligned with region of interest and top-level function, provisional creation of hypothetical component diagram and the functional realization graph is conducted. I will explain the FRZ in the next slide. And after construction of HCD and FRZ is conducted, rejecting diagram that are inconsistent with another scientific knowledge. So to build software that functions like the brain, it is effective to use reverse engineering, which creates a functional hierarchy diagram by concurrently employing both bottom-up design, like building up functions from parts and how finer components achieve specific functions. And top-down design, breaking down functions from the whole, how the software's parts of the whole achieve specific function. The SCID method is a design methodology based on this approach. FRZ is a functional hierarchy diagram that represents how upper-level nodes with TLF as a top node, and realized from combination of functional groups at lower nodes. The nodes at the lowest level of this functional hierarchy represent the functions of HCD components. An important point is that these bottom-level nodes correspond to brain structure known as BIF, so the components. And you can check the data and papers from the BRICE repository, but for this presentation, we have provided a link to the workshop website. Please close the website and click on data and paper to check. You can see the BRA data image file and data paper if you access the link. Okay, here is the BRA data. And I explained the BRA data structure using YM24 amygdala that is published last year as an example. This projected seed, which opens first upon access, provides an overview of the data. This seed contains information about the contributors who create the BRA data, and details such as the project ID in BRA data particularly. And circuits, connection, and FRZ are important seeds, so we will explain these seeds. Okay, the information necessary for understanding BRA data is constrained in the following three seeds: BIF seeds, and HCD and FRZ seeds. The circuit seed and the connection seeds and FRZ seeds. The circuit and connection seeds contains the BIF information, and that is anatomical data, and while the FRZ seeds contains HCD and FRZ information. Okay. The circuit seeds contains information on the neural nuclei, nuclei groups, and the cortex within the ROI, as well as their anatomical hierarchy relationship. The data are below the yellow line at the bottom of the seeds here. You can see the castle. The data below the yellow line at the bottom of the seeds are pre-registered whole BIF This data is registered based on the anatomical structure recorded in the Allen Institute's DHBA. Contributors, while referring to whole beef, register new circuits as needed. For example, the low-level INADM indicates that there is a circuit with the ID INADM within the ROI, and various information about the circuit is registered. As noted in the names column, these circuits represent the dorsal cluster of medial ITC. The existence of the circuit is demonstrated in Hagiwara 2021. INADM is a group or graph order number 108024 in the DHBA, which represents the ITC, intercalated nucleus of amygdala, and it is positioned as a subcircuit of this ITC. This INADM is a uniform circuit and consists of GABAergic neurons. And this is a connection sheet. The connection sheet registers connections between circuits listed in the circuit sheet. In this example, a projection from INADM to INAVM is registered. Incidentally, this projection is shown to be supported by anterograde tracing experiments in previous research, Hagiwara 2021. And this is a FRC sheet. The sheet registers what function each circuit performs and how the combination of these circuits achieves the top-level function. For example, in this law, I showed that by combining a circuit called PA peer with a capability called relay function, it achieves a function of input normalization. Next slide, I'll move on to the next slide. And okay. This is a BLA image. Next, I will briefly share about BLA images. This consists of BIF image and ACD images and FRC images, each representing BIF and ACD and FRC. In diagonal form, these images serve to make the content of BLA data easier to understand visually. Okay, so how to open BLA images with Draw.io. This is a brief introduction. Please access the data and paper from our workshop website and click the XML file and open with Draw.io. Please note that you need your Google account, and then you can open it in the PNG file. So this is the last slide. I will explain the BLA data, and you can also see the paper on our website. The BLA data paper provides a linguistic explanation of the content described and explains how the data was created. The data paper also serves to supplement the description of the BLA data, and these BLA data are evaluated from three perspectives. This is three perspectives: comprehensibility and reproducibility, transparency. So comprehensibility means the contents of the paper is clearly and concisely written, making it easy for readers to understand. And reproducibility means a method for data collection, processing, and analysis are described in detail, allowing other researchers to reproduce the same results. And finally, the transparency means the process of constructing the dataset, and tools and techniques used are thoroughly explained, making the entire research process clear. Okay, this is my last page, so we would like to move on to oral presentation. Okay, so thank you. And so the oral presentation is three presentations. Each presentation is 15 minutes per talk and 5 minutes for Q&A. But second presentation 20 minutes talk. So first speaker, Taniguchi Sensei, can you share the screen? And if you have, okay, please move on the presentation.
はい、Good morning. I'm Akira Taniguchi, Ritsumeikan University. I'd like to talk about data of brain reference architecture of AF24 hippocampus amygdala, integrating BLA for spatial cognition and fear conditioning. Here is abstract in this talk. Firstly, hippocampal formation plays a key role in spatial cognition and episodic memory. And next is amygdala is essential for adaptive fear conditioning. And the brain reference architecture BRA data was developed by integrating amygdala BRA data and hippocampal formation BRA data here. And it's that enhanced previous models by incorporating new brain information flow BIF, which map connection between the hippocampal formation and amygdala. BIF construction, the contribution is we provide foundation for understanding high order function related to spatial cognition and fear conditioning. And expected benefits. Firstly, improved understanding. It's clarify the anatomical structures and functional interaction between hippocampus and amygdala. And next is deeper insights. It's enhanced knowledge for each regions and each region's roles and how their interaction influences complex cognitive and emotional processes. The two BRA data of hippocampus and amygdala were reported at the first International Whole Brain Architecture Workshop. This link is here, and they were then published on BRA-ES, like this here. The two BRA data were constructed separately and not integrated. We adapt the BRA-driven development methods that structured inference decomposition, SKID methods. BRA-driven development is methodology for building software based on the brain architecture. A SKID method was used to design software that was consistent with the brain structure and function as obtained through neuroscience knowledge, like this. Here is this talk's context. This data paper reports BRA data on spatial cognition in hippocampal formation and fear conditioning in amygdala. BRA data is usually limited to individual areas with specific function, but to construct a whole BRA, it is important to construct BRA data for the connections between different areas. This is the first attempt to integrate BRA data like this. Then I talk about the hippocampus. Hippocampus is brain region related to the spatial memory in animal and deeply involved in spatial learning and spatial exploration and so on. Their hippocampal formation is core areas for cognitive processing. In neuroscience and cognitive science, knowledge has accumulated on how hippocampal formation contributes to spatial cognition of organisms. So it will become essential building blocks for construction of integrated BRA and whole BRA. Hippocampal formations neuroscientific knowledge is very useful. This is the neuroscientific findings from the hippocampal formation. Main function are navigation, cognitive map, self-localization, and path integration. And the hippocampal formation plays an important role in navigation and cognitive map. In addition, some neural cells related to these functions have been founded. It's, it believes the place cell and grid cell involves in execution of special cognition processing. In, in this figure shows the brain information flow diagram for hippocampus formation based on anatomical structure. And region of interest is here, and input-output is here. Top-level function are self-localization and place categorization by integrated allocentric and egocentric information. This diagram shows the hippocampal formation based on probabilistic genetic models on brain information flow. Allocentric visual information from MEC and egocentric visual information from LEC are integrated in the hippocampus. And right circuits here, this related to the self-localization, and the left circuit is related to the visual representation of place. Based on that HGCD PGM, we constructed a function realization graph in hippocampal formation. This figure is FRG graph. In summary, the top level function is spatial cognition, which is supported by spatial place categorization and self-localization. Spatial categorization is primarily supported by LEC area, and while self-localization identification is mainly supported by MAC area. These functions are integrated by the hippocampus. Next is amygdala. The amygdala regions closely associated with the fear condition, including LABA, CEN, INA areas. Fear conditioning is when incurious conditioned stimulus is paired with a mucous and conditioned stimulus, leading to fear response to a previous incurious conditioned stimulus. This process is modulated by the hippocampal CA1 region, which is process contextual information to ensure that fear responses are appropriate to the specific context. This figure shows the base of the BRA data in amygdala. Here, LA region receives sensory information, CS and US, and facilitated Hebbian conditioning, and LA project to the BA region contains BA fear and BA EXT. And BA project to the CEM, which is involved, including the fear response. And INA and CA1 serves as a modulator of the BA. Right figure shows the amygdala's BIF data, BIF diagram. In this paper, we have developed it as a BRA data format by integrating this amygdala BIF and hippocampal formation BIF. This is list of abbreviation of the fields used in this BIF. Please refer the data paper for the details. Here we will describe the this figure is constructed integrated BIF figures. Here we will describe the circuit added to the BI data. Firstly, CI context in amygdala BRA data was changed to CI1 ventral. Then CI S ventral, subiculum ventral was added. Next is in hippocampal formation BRA, CA1 distal and CA1 proximal in hippocampal formation BRA were defined as a subregion of CA1 dorsal. And same way is subiculum is same way. Next is describe a connection added to the BI data. Firstly, CA1 ventral was known to project to BA FIA and BA XT. This projection. And there is projection from BA1 to CA1 ventral. Here. Similarly, S ventral has a bidirectional projection to BA1. And CA1 ventral has projection from CA3 and LEC 3 layers. And C3 is here, LEC 3 is here. And MEC 3 layers has projection to CA1 ventral and S ventral. It's this line. And from CA1 ventral, there are projection to LA and S ventral and S dorsal and MEC 5 layer. There is also projection from S ventral to MEC 5 layer. And LA, FIA, and BA in amygdala and LAC2 and 3 layers have bidirectional projection. These lines. And BA has projection to CN3 ventral. Finally, it have been suggested that prefrontal cortex and BM within amygdala are important for the fear conditioning and elimination. Therefore, we adapted projection with PFC and updated version of amygdala's BIF. Towards the construction of functional hypothesis, one suggested two functional different pathway between BA in amygdala and hippocampus area. That's direct and indirect pathway. The indirect pathway is here. which process through the LEC with likely carries integrated semantically rich emotional information that's including this recent experience and environmental elements. In direct projection, direct pathway, it plays the role in meditating fear conditioning learning here. And about ventral and dorsal hippocampal formation, Xian and speculum. Spatial information is related to proximal area and non-spatial information related to distal area. The ventral area is mainly connected to the amygdala and is responsible for processing related to anxiety, goal directedness, fear, and sociality. The dorsal area is responsible for spatial processing compared to the ventral area. And about PFC and HF hippocampal connection. Also, this is outside of this RY is this VRA. There is also pathway between hippocampal and amygdala that goes via the ventral medial prefrontal cortex. However, Park reported that cognitive control in an active place avoidance task is not dependent on MPFC damage. Here's summary. We have developed the BR data BIF format by integrating amygdala and hippocampus BR data. This BR data expands on previous BR data by incorporating new brain information flows that capture the connection between hippocampus and amygdala. This study investigates that multiple pathways connect the amygdala and hippocampus. The BIF of hippocampal formation of amygdala were integrated connections between ventral and dorsal hippocampus and an indirect pathway through connection between amygdala and LEC mainly. Finally, by constructing some computational models for this BIF, we will able to realize emotion adaptive navigation to implement to the mobile robot. This is next step. That's all. Thank you for listening.
Thank you for your presentation. Do you have any questions or comments?
Thank you for your presentation. And so I may have missed some points and I may have missed the overview of your research, but so you talked about the types of information that flow through hippocampal formation or amygdala formation. And I'm wondering whether you also incorporated these types of how can I say the types of information that flow that you've incorporated into your models, the types of information you assume to flow within these hippocampal formation or in amygdala formation.
Firstly, my we 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 have worked a previous study about the hippocampal formation BIF data, and another groups have constructed amygdala BIF data, and I try to construct BR data from integrated BR data. It is my interesting point, but yeah, sorry.
I have an additional comment about that. Now is the steps of the just making the BIF is the anatomical structure. But in the BRA development, we have built after that HCD and FRD that one related to the functional. So in that designing stage, we are rethinking of the already なんていうか stated something kind of function is more plausible or not. So we are going to think about the next step. So think about what meaning is a signal is interact with the hippocampus and amygdala. So anyway, next step.
It's not yet incorporated. I mean, I think it's more reasonable. I mean, I agree in that sense that it's more reasonable to not really assume what type of information is there at the stage of building.
That's now referring the previous work.
Okay, thank you.
Okay. Oh, can I ask? Then it's.
Oh, so now the time is up. So thank you for your presentation, Taniguchi-sensei.
Thank you.
So next presentation is by Naohiro Yamauchi. Can you hear me? Okay.
Yes.
Please share screen, and if you have ready it, please go ahead.
Yes. Could you see my screen? Yes. Thank you for giving me this great opportunity to present my work. So the presentation title is Data for Brain Reference Architecture of NY24 Canonical Cortical Microcircuits Inference and NY24 Canonical Cortical Microcircuit Decision Making. I'm Naohiro Yamauchi from OIST. So our focused brain region is canonical cortical microcircuit, CCM. So the neocortex is largely developed in humans and important for enabling intelligence. So this is our cortex, and actually neocortex has involving many functionality like visual processing, auditory processing, and motor processing, and also decision making and emotional things, and so on. But important thing is for microcircuits of the neocortex, this 6-layer architecture of CCMs are preserved throughout the cortex, and it's repeated 100 million times in the cortex. So it is thought that the CCMs are computational unit of our cortex. So let's go to the anatomy of the CCMs. So my project main focus is the canonical cortical microcircuit as a brain region, but we also model the input or output from the higher other area of the cortex, lower cortex, thalamus, and basal ganglia, and ventral tegmental area. And also I model the 6 layers of the cortex. The main sequential information pathway in the cortex is from thalamus to layer 4 to layer 2, 3, and layer 5. But this is kind of a classical view, and recent studies shows that there are many other connections, and its anatomy is more complex. And for anatomy, another important thing is the cell types. And for main excitatory cell types, there are intratranspheric neurons, pyramidal tract neurons, and corticothalamic neurons. And for inhibitory cell types, parabolomin neurons, somatostatin neuron, vasoactive intestinal peptide neuron, and neurogliaform neurons are main cell types in CCMs. So because of the importance and the universality of the CCM, various hypotheses have been already proposed for the computational function of the CCMs, such as Bayesian brain hypothesis, predictive coding, and active inference. So this is kind of the summary table, and each column represents each publication proposing the computational model, and each row corresponds to the uniform circuit. And each cell represents the output semantics from the circuit. So actually, for example, many models assign the output semantics prediction to the layer 2, 3 neuron, but some studies assign the prediction error. So these models are somewhat consistent, but somewhat inconsistent. So purpose of our project is build a CCM's model that is consistent with the detailed neural anatomy and enables cognitive inference and decision making. So in our project, we constructed the anatomical data, PIF, and functional data, HCD and FRG. And you can find the data in this PRD Literacy System URL. Okay, so this is the outline. I first talk about the anatomical information, brain information flow, and then I talk about the functional information, hypothetical component diagram for inference, and hypothetical component diagram for decision making. So as already Tauchi-san explained, I follow the scikit method developed in the WBA. So in this approach, I first register anatomical data of CCMs and construct brain information flow, and then propose computational function of CCMs and construct CCM's inference model, hypothetical component diagram for dynamic Bayesian inference and controllers inference. So this is about the anatomical information sampling strategy. So this is kind of the summary table of the anatomical references I cited. So each column corresponds to each literature. Some are reviews and others are modeling paper. And each row corresponds to each uniform circuit. So you can see that some literature summarize the layer 2, 3, but other literature separate the layer 2 and layer 3 IT neuron. So there are variability. So we made a comprehensive and detailed anatomical information by integrating each study. And hypothesis is that if the circuit in the reference was coarser than the uniform circuit, all connections were assumed between subcircuits, which follow the PRA data development strategy. So this is the final brain information flow I developed. So each row corresponds to each layer. And in the left side, I summarize the excitatory neural population, such as layer 2 IT neuron and layer 3 IT neuron. And in the right side, I summarize the inhibitory neural populations such as SST neuron and VIP neuron. And solid lines represent the excitatory connections, and dashed line represent the inhibitory connection. So it's quite complex. And next, I explain the functional part. So I follow the reverse engineering approach of the brain. So identify plausible hypothesis about the computational function of the brain, and use both top-down and bottom-up design approach. In the top-down design approach, the breaking down the required function to obtain a structure to achieve top-level function. On the other hand, in bottom-up design approach, build up a structure by stacking implementable function from each uniform circuit. And especially in bottom-up approach, network motifs, concept of the network motifs is important. So network motif is the small recurring pattern of interconnection in the network. So in this review paper, in the cortex, inhibitory, it's described that inhibitory neuron function as a component within the network motifs. So this feed-forward inhibition, feedback inhibition, and this inhibition is the main network motifs. And for example, the thalamocortical feed-forward inhibition is the PV neurons are involved, and also VIP somatostatin interneuron mediated disinhibition and feedback inhibition involving the PV and somatostatin interneurons. And for the specific algorithm, dynamic Bayesian inference was chosen as an algorithm that could be implemented in the canonical cortical microcircuit to achieve cognitive inference. What is the dynamic Bayesian inference? The dynamic Bayesian inference is sequentially estimating uncertain time-varying internal states based on the noisy observing data. So I denote the observation as O and state variables as S. And this algorithm assumes two conditional probability distribution, dynamics model and observation model, and use the previous posterior prior and dynamics model to compute the predictive prior, and combine this to observation model with new observation to update the posterior prediction of the internal state, which is the goal of this algorithm. So this is the brain information flow I already explained. And based on this, I developed the hypothetical component diagram, which use the subset of the P connections and compute the each variable using the message passed by the each connection, and finally compute the posterior prediction. And another important component of the PRA data is the functional realization graph, showing hierarchical dependence between the functional nodes to achieve the top-level function. In this case, cognitive inference from circuit components. So this graph shows that the cognitive inference are hierarchically achieved, like the prediction, likelihood, and posterior, and then circuit motifs, and then finally from the uniform circuits. So briefly, the cognitive inference can be divided in three parts, like calculate likelihood with observation model, and update posterior with dynamics model, and calculate map estimation, maximum a posteriori. So yeah, let's explain the relationship between the HCD and FRG more detail. So first, I explain the this circuit motif, the winner take all. So winner take all is achieved by the feedback inhibition circuit motifs, and in this review paper, somatostatin neuron involved this circuit motif, and I assume that this layer 5 BT accessory neuron and layer 5 SST inhibitory neuron bidirectional connection enable this winner take all circuit. And then in a more higher order, I assume that posterior prediction update is calculated. This is the equation of the posterior prediction update. So this need the 3 terms, and this modulated sensory evidence passed from the layer 2 IT neuron, and this purple predictive prior information passed from the layer 5 IT neuron, and this normalization term is achieved using this feedback inhibition circuit motifs I already explained. So yes, this is just an example of the correspondence between the FRG node and the HCD, but this kind of relationships are is in the each node of the FRG and the HCD circuit. Then I explain the function of the decision making. So in the computational field, the duality of sensory inference and motor control is known for the controller's inference. Yeah, so well, intuitively, for example, if I grab the bottle, we have to infer the weight or shape of the bottle. So in this sense, the inference and the motor control is somewhat connected. But this controller's inference mathematically describe the duality of the Bayesian inference posterior distribution update and reinforcement learning value function or value update. Yeah, the reinforcement learning is the algorithm in which the agent seek environment and learn the optimal policy to get the maximum reward, and sometimes used as a model for decision making. So in more detail, controller's inference interprets control as a task for maximizing the probability of desired trajectory through a joint distribution of a state and action. And dynamics model and reward function are assumed, and action value is calculated from reward based on the state transition model and state value. And state value is calculated by taking weighted average of the action value. And finally, by subtracting the state value from the action value, advantage or policy is computed. And important point is that we start building HD from the completely same PIF image, but different HCD hypothetical component diagram for cognitive inference and decision making. And for cognitive inference part, I already explained. And for decision making, I made this diagram and in which state value and action value is computed and which is used to calculate the policy. So this is the FRG for decision making, and in briefly, decision making can be subdivided to the calculate state and action values with reward function and predict state with dynamics model and compute policy based on values. And this is the some consideration for the data usage. For brain information flow, projection to inhibitory neurons are underrepresented in the previous literature because of the limitation of the experimental technology. So in this sense, the artificial information is still not complete. And for the hypothetical component diagram, so yeah, the HCD only account for a subset of if connections, and many connections are not used. So still, this is the hypothesis about the functionality of the CCMs. And finally, for top-level function, the present HCD is limited to the function related to the inference and decision-making, but our cortex may do many more various things. So these are the development of other HCDs are future direction. So for conclusion, we outline how cognitive inference and decision-making could be achieved through specific excitatory neural population and circuit motifs involving inhibitory population within canonical cortical microcircuits. So this is summary about the semantics for each cortical excitatory neuron types, and each column corresponds to the dynamic Bayesian inference and controls inference output semantics I model. Yes, thank you very much for listening, and these are the references I used for this study. Thank you very much.
Thank you for your presentation. Okay. Oops. So if you have questions, comments?
So, it seemed to be focusing on, the cortical region. So how do you think about the subcortical and cortical relationship or the difference between the cortical region, motor cortex or visual cortex?
Yes. So thank you for the question. And first of all, I current study I focus on the cortex, only cortex. Yeah, but sorry. Yeah, I also somewhat model the input or output from the VTA and thalamus and basal ganglia. So somewhat I also model the information from outside or information toward the other brain region. But I think a more detailed relationship between the other brain region is the future direction. And for the second question, yes, that's true that I only model the inference and control, so to function. And yeah, we know that there are many function cortex too, but briefly we can divide that anterior part of the cortex involving the motor function like the control or decision making, and the posterior part for the visual and auditory, but still it's abstractly the inference, sensory processing and related to inference. So yeah, I think this our model may be related to many functions still.
Okay, thank you very much.
Do you have any other comments or questions?
Can I ask a question online?
Okay, please.
Yeah. Thank you for your presentation. So you have gone through quite a large number of papers, and then first summarize the data in Excel file, and then created BIF and then SCD and FRG. So in this process, how much work was done just by hand, or how much it was automated or with any computational assistance too?
Yes, yes. Thank you for the question. Yeah, I think maybe the WBA manager Taotsu-san or Yamakawa-san were more familiar with, but yeah, in my WBA also developed a kind of automation system which accumulate the information from the papers and also some automated function in a spreadsheet to automate some process. Yeah. So yeah, this work is kind of the combination between this system. But yeah, for this canonical cortical microcircuits, actually there are many previous good reviews and modeling paper, and so already kind of the information is somewhat well accumulated, summarized. So actually doing manually integrating this information is easy compared to other brain regions, I think. So yeah, for this study, the manual compilation of the information is, I think, main compared to the automating the information compiling. Yeah, but I think for other brain region, I think it's important to automate the process, and I think that's what WBA aims to do.
Okay, yeah, thank you.
Yeah, thank you very much.
I have one. We can take one short comment. Sorry. Yeah.
Thank you for your talk, and I'm wondering, no. So can you describe a little bit more about why, more background or motivation about why you compared the two types of hypothesis or the theories to model the CCM architecture?
So you mean the dynamic Bayesian inference and contrast inference, right?
Yes, that's right.
Yes. So the general motivation is that because there are many functions, and and basically Cortex can do both motor control and sensory inference using this similar anatomical structure, and this is somewhat kind of mystery, but in the computational field, it is known that sensory inference and motor control or decision making is connected using this kind of theoretical framework, the duality of sensory inference and motor control. So maybe this theoretical framework is implemented in the Cortex. That's why Cortex can do both sensory inference and motor control using similar anatomical structure. That's our hypothesis. And so then from the same anatomical structure, I developed the sensory inference function and motor control or decision-making function, which has correspondence from the theory, and propose the model. That's my motivation.
Thank you for your presentation. So now is the time to finish this presentation.
Thank you very much.
Okay. My presentation is last in this session. So I would like to talk about the data for brain difference architecture of YT24ITC and AIMY. So I'm Yoshimasa Tatsuji from the University of Tokyo and WBAI. So I would like to introduce my interests. I like interest such topics, the uncanny valley. If you have heard before, I would like explain before I explain the registration of the BRA data. So the concept of the uncanny valley was first hypothesized in 1970. Generally, when robots represent resemblance to human is low, people do not feel much affinity toward it. And as the robot's appearance becomes more human-like, the sense of affinity increases. However, once it becomes extremely similar to human, that affinity actually turns negative. It is said that this gap is particularly pronounced when the robot is in motion. Okay. The recent study showed real issue may lie in the fact that these visual stimuli from the perspective of human categorical perceptual characteristics located at the category boundary because they are so ambiguous stimuli between the human category and the robot category. So it becomes difficult to judge which category they belong to. And this ambiguity in turn generates negative emotions. Psychological experiments shows for facial images located at this category boundary, the time response latency required to determine whether they are human or not is shorter than for facial images whose category is clearly defined. Okay, so aim to given the background and my own interests, I created BRA data related to human face perception. Faces are extremely important social signals for social animals, including humans. And it is also known that various brains are involved in face perception. Therefore, as first step, I believe that constructing BRA data concerning face perception will serve as a foothold for developing more advanced BRA data on face perception in the future. And furthermore, BRA data can be considered a blueprint for brain-spiral architecture that realize complex cognitive functions. Building an agent capable of identifying human faces based on this blueprint BRA data could also contribute to symbiosis between humans and brain-based agents. So in the BRA data, the main brain region of focus inferior temporal cortex and amygdala. Both of them are very related with face perception. And assuming that these regions or perform familiarity-based categorization, the BRA data was designed accordingly based on this top-level function. So brain difference architecture has 3 steps to construct BRA data. So investigating neuroscientific paper and data, and design the BIF, which means anatomical structure, circuits and the connections. And then design the HCD, FRZ, which means functionality of the brain regions. In this study, which means inferior temporal cortex and amygdala. So for I would like to show about the brain regions related to face perception in inferior temporal cortex, which subserves high-order visual processing and which subserves view invariant face processing. And also known, it's neural populations in the inferior temporal cortex has global to fine processing, which means for the example, this figure shows global to fine processing in the inferior temporal cortex. These are
neural activity of the neurons of the inferior temporal cortex. And the first, all of them show it's activated when the face is shown. Then all neurons in the inferior temporal cortex activate for the first steps. But actually showing individual too, these neurons spontaneously keep on activating, but the other does not. So which means these all of the neurons in the inferior temporal cortex showing in this figure activates firstly activate when the face is shown, and then the individuality individual activates certain population. And the next brain region is amygdala. Amygdala is also related with face perception. Amygdala is also introduced by the Taniguchi sensei, but I would like to introduce it. And it is almond-shaped nuclei in the temporal cortex, and subdivision is lateral, basolateral, central nuclei, and so on. It is well known to respond to various stimuli, including face. But major activation is well known is to the negative valence of facial expression, like fearful faces. But recent study shows that emotionally neutral face is also activate the amygdala. And the amygdala regions deficit preference on looking face-like stimuli. So these are showing amygdala has a very important role to face perception. And the circuit and the connection is well investigated, and the lateral and the basolateral amygdala are key subdivision of amygdala and related, especially related to ITC. Basolateral amygdala has both excitatory neurons and inhibitory neurons. And ITC, inferior temporal cortex, and amygdala are interconnected. TE and TEO is a major part of inferior temporal cortex, which projects to lateral basolateral amygdala, and also feedback from amygdala to inferior temporal cortex. So TE and TEO are projected from lateral, basolateral, and medial nucleus of amygdala. So I showed BIF data, which means anatomical structure of inferior temporal cortex and amygdala. So I registered 5 uniform circuits as a ROI. So the table and the figure on the table. So square shows uniform circuits, and the rounded square shows the circuits. So which means groups of uniform circuits. And the circle shows the inputs and outputs. In this figure, there is only input. Before you see means a uniform circuit and before area. And PUL means pulvina in the thalamus. So the before you see project to inferior temporal cortex excitatory neurons, and then project to basolateral nuclei inhibitory neurons of the basolateral amygdala. And the pulvina is also projects to the lateral part of the basolateral of the amygdala, and the lateral part of the basolateral amygdala and the basolateral inhibitory neurons in the basolateral nuclei projects to excitatory neurons of the basolateral. Then the also basolateral nucleus projects feedback to the inferior temporal cortex. So the this connectivity is shown in this figure. And then inferior temporal cortex excitatory neuron and the inhibitory neurons of the inferior temporal cortex has interconnected. These are registered in the BIF data, which means so I registered ROI of my project, and it includes these subcircuits, and then they are registered in this column. And also, BIF data has a connection state. So I registered 8 connections here. So I explained it, already explained. And for example, this blue circle means excitatory neurons of ITC to the inhibitory neurons in basolateral of amygdala shows this blue line. And red and green is also registered in this style. Okay, next I would like to explain about the functionalities of the inferior temporal cortex and amygdala. And functionality of the ITC is investigated with the computational neurosciences, and dynamical model has been proposed. So this is a complicated problems and a dynamical system, and this model shows that reproducing the behavior of inferior temporal cortex neural populations by recalling vector patterns stored in the attractor network. So, which means I define the functionality of the inferior temporal cortex can be calculating the stored memory pattern at time t in the inferior temporal cortex, excitatory neurons, from signal modulating memory pattern from the inferior temporal cortex in the inhibitory neurons. And I would like to explain about the important functionalities of the amygdala. So amygdala is well known in the face of perception and fear detection or something, but response to not only fearful but also emotional neutral faces. So according to the Sander in the 23, I would like to accept the hypothesis of relevance detection. Relevance detection is of the kind of the function of the amygdala, which is amygdala. It is involved in the detecting the biologically important signals. So I suppose the amygdala has a function of the relevance detection. And this is overview of the data. So I will not explain about the detail in the data. Please refer the data in the this QR code. And I will explain about the overview of my HCD and FRZ data. So designing HCD and FRZ. So of course, HCD should be aligned with BIF, which means anatomical data. And so HCD is similar with and identical to the BIF data. And the 3 sub-function that achieves top-level function. Top-level function is the familiarity-based categorizing. So I defined the 3 part of functions. I would like to explain that these 3 sub-functions. So blue part is function which is realized by the inferior temporal cortex. Inferior temporal cortex is association. So as the previous study shows. And the red part is relevance detection capability of lateral portion of the basolateral amygdala, which is here. So Palvina is encoded the visual stimuli. Then the lateral part of the basolateral amygdala can detect the relevance. And the green part is BLEX, which is 2 input BL inhibitory neurons of the basolateral and the lateral part portion of the basolateral amygdala, which is anatomically connected. Anatomical connection can form error detection motif. So the 2 inputs from the Palvina and the inferior temporal cortex excitatory neurons, which is calculated error detected. So Palvina, if Palvina shows it's like a relevance, like a face, but the inferior temporal cortex does not code it that is not face, then the error detected in the basolateral amygdala. So this is a hypothesis of our BRA data based on the SCID method. And whole part in this inferior temporal cortex and amygdala, the orange part shows calculating the association signal of stored memory pattern modulated by relevance from visual input pattern. Visual input pattern has two types, and V4 uniform circuit and Falbina. So this suggests that ambiguous phase stimuli may cause error increases and lead to delays convergence of ITC neural dynamics. And this is all part of the BLA data which I created. And after that, I simulated and based on the computational model. And this is the results of the simulation. Briefly introduced. And if the person's pattern vector and the robot pattern vector is merged, and so which means make ambiguous phases and show to the inferior cortex, which this is a pattern of the activation pattern. So dotted line in the figure shows overlap person 3 pattern. So if the dotted pattern is high, the activation pattern is, or it is human. But on the other hand, the how to say dot end line, and this line shows the overlap robot 2 pattern. So which means the same input, but the robot 2 pattern is higher. So its unstable pattern were observed. So firstly, this ambiguous pattern is it means like human, but it's not a human, and then it may be robot and human. So that is very unstable pattern were observed based on the BRS data and the simulation. And also, this is another simulation results. Input is just a robot pattern. Then the inferior temporal cortex pattern is just like, it's a robot. So there is that the human does not present it. So it is no problem. Unstable pattern cannot be observed. Okay, conclusion. Sorry for not constructed presentation, but I would like constructed BRA data of the inferior temporal cortex and the amygdala. But the inferior temporal cortex is just a 2 uniform circuit in the excitatory neurons and the inhibitory neurons. So only 2 uniform circuits is adopted. But as everybody knows, neocortex has 6-layer structure, and Fujita has investigated the microcircuits of ITC. So I would like to more precise BRA data. Okay, that's all. Thank you for your attention. And if you have any questions and comments.そうです。
自分のズームで入ってもらうと思う。自分のズームで入ってカメラは。
I have a question about the regions inferior temporal cortex, and I just a mistake to understand what is your final goal of the functions. So, for example, decision making, or so in this case, why do you focus on inferior temporal cortex only? Yeah.
So why I use inferior temporal cortex is just like there is many computational models well investigated. So as you said, pointed out, there is many brain region which is plays key role to face perception. Like okay, this is face is related to the decision making, and that is like a best career or something. But not my preference. But yes, but in this stage, I just only the inferior temporal cortex. And the inferior temporal cortex is very key portion of the face perception and related with the uncanny valley. So I use this. Why is amygdala? Okay, amygdala is also important place to face perception, and there is no computational model as far as I investigated, there are combination of the inferior temporal cortex and amygdala. So I adopted to keep part of the key region of the face.
Okay. Thank you.
Thank you.
Hi.
Okay.
So I have a question about the FRG.
Yes.
So in your please show the FRG.
Okay. Yes.
Here.
Okay. Here.
In this FRG, so you don't include the LDL
LDL.
in the same part of the error detection.
Yes.
Why that is separate?
Okay. That's important point. But yes. Actually, I consider this because it may include in the this part. Yes. So you pointed out. why this-
That red
Red
part is very completed with DLEX.
Yes.
So we can make some cluster of the network,
Yes.
subcluster. So in my feeling, it is natural, including these three uniform circuits into the one sub-node.
Yes.
That is my question.
That's point. Actually, yes. So I should reconsider about the qualification of the FRZ. So I would like to discuss with later. Thank you. Maybe other questions? Other questions? Okay. So maybe just is the time. So okay, this I finished my presentation, and thank you for your attention. And okay, I would like to close this session, and we have the break for 10 minutes. Then like 10:50, we would like to start the panel discussion. Okay, thank you for your attending. Okay, please break. Thank you. 50分から先生。
So, in the beginning of panel discussion, so I want to provide some topics, and this is my agenda of today's my talk. First, I want to self-introduction. For self-introduction, I present my research in the previous lab and in our lab. And next, we like to show our BRA, BRA on the vesical ganglia. And next, atlas, I'd like to show provide some topics. So first, I would like to introduce our works in the previous lab and the independent lab for my self-introduction. In the previous lab, we investigated motor learning. In everyday life, we accomplish our goals with skilled movements, such as piano, dance, and key typing, and even dishwashing. To evaluate neural activity during learning and execution of intentional movements, Matsaki Lab has established a self-initiated lever pull task of head restrained mice. Mice were trained to pull a lever with a right forearm to get a reward, and they learn this movement. And this is a really simplified schematic for brain circuits for motor learning and execution. These cortical and subcortical motor structures are known to be important for movement. Injecting AAV expressing TD tomato into the thalamus visualize thermic axons in M1, as you can see, densely in layer 1, also deep layer 2, 3, deep layer 5 and layer 6, and focal retrograde tracing from layer 1 of M1, stained thermic nuclei, including VA and VM, and known as basal ganglia-related nuclei. By contrast, the bolus retrograde tracing from layer 1 to layer 5 of M1, additional staining was observed in the VR nucleus, known as the cerebellum-related nucleus. These results indicate that VA and VM neurons project their axons to L1, and VR neurons mainly send axons to deep layers. We next tested whether these thalamic axons are potent to evoke the limb movement. The primary candidates of subcortical input to the motor thalamus are basal ganglia and cerebellum. So we confirmed anatomical connection from substantia nigra reticulata (SNR) and or deep cerebral nuclei (DCN) to the motor thalamus. And SNR is an output of the basal ganglia, while DCN is an output of the cerebellum. So, and as you can see, axonal arborization from SNR and DCN in the motor thalamus are mostly distinct. And then we photostimulated SNR or DCN. And so we investigated functional connectivity from SNR to or DCN to M1 via the thalamus. We performed the optogenetic activation of SNR or DCN, while simultaneously thalamocortical axons were being imaged. So by photostimulation of SNR, a rebound excitation was detected, like these traces, and this is consistent with inhibitory nature of SNR neurons. On the other hand, by photostimulation of DCN, immediate excitation was detected, like these traces. So as you can see here, by photostimulation of SNR, layer 1 buttons were activated more than layer 3 buttons, and vice versa. The motor thalamus provides principally distinct pathways from the basal ganglia and cerebral signals to the motor cortex. So we here show axonal activities of the primary motor cortex in vivo by 2-photon calcium imaging. These are time-averaged images of layer 1 and layer 3 in early and late sessions. We determined as many ROI as we could take by a semi-automated algorithm. The lever trajectory and leaking is shown here. And these blue and red traces are the calcium traces. Note that the calcium activity was steadily recorded even when the mice's body moved with vapor, thanks to offline 3D motion correction. And the dynamics of thalamic activity is shown here. And here are the trial average activities of thalamocortical axons during lever pulse. And this is lever traces and leaking traces and reward timing. And these activities are aligned at lever pull initiation and initiation or termination. And so it's a lever pull initiation aligned activity, and it's a lever pull termination aligned activity. The population average of activity changes rather slowly in layer 1. However, the slope of mean activity peaked in time of the initiation and termination of lever pull in late sessions. This means that the population of thalamic axons is synchronously activated at that time. And these are histograms of the peak timing. In late sessions, layer 1 thalamocortical activities exhibited start and end activity during lever pulse. In contrast, layer 3 thalamocortical activities exhibited only start timing during lever pulse. And thus layer 1 neuronal activity, layer 1 thalamic activity is shows distinct sequential activity. So we quantified the sequential activity in layer 1 and layer 3 thalamic axons, and layer 1 shows the longer sequential activity. And I skipped. I want to skip the detail because of time, but we analyze in statistical method, and we found L1 thalamocortical activity is related to lever kinematics, and layer 3 TC activity is related with success rate. And these difference is obviously because of the origin of information, and the layer 1 activity is origins from basal ganglia. So the in independent lab, now we are trying to assess dynamical interaction with cortex, interaction of cortex and striatum. So, and now analyzing neuronal dynamics and information flow in cortical basal ganglia, thalamocortical circuits. In biology of the behavior change, we are aiming to develop that behavior task useful for dissecting brain dynamics. And we also would like to use imaging techniques such as 2P microscopy and 1P wide field microscopy with neuropixels. And we intend to record neurons in cortical layer 2C or layer 5A with imaging neurons in deep layer in the cortex and in deep structures such as striatum and thalamus with neuropixels. So this is our lab, and this is an imaging guy, Ryo Aoki, and he is and Shoya is a very good task builder. And the work in previous lab is mainly done by Yasuo Tanaka and under the supervision of Dr. Matsuzaki. So next, I'd like to move to the next section. So with a curiosity about the principles of basal ganglia circuit, I am making BRA on the basal ganglia with Dr. Namb from NIPS in Japan. And based on many experiments, Dr. Namb wrote review papers. We are now making BRA on the basis of these review papers. The basal ganglia from loop circuits with the cerebral cortex and play a crucial role in motor execution and learning. Now we are investigating the connections mainly between motor basal ganglia and related regions and converting them into the brain information flow. Also, we are considering releasing appropriate motor output at the appropriate time corresponding to cortical motor commands in physiological conditions as a top level function in the FRG of the basal ganglia circuits. And these are typical dynamics in GPI SNR. This is a basal ganglia output, and we in short, we like to reproduce these activity dynamics of basal ganglia output. Also, and we intend our BRA of the basal ganglia to reflect pathophysiological network changes in movement disorders. The malfunction of the basal ganglia causes severe motor disabilities observed in movement disorders, such as Parkinson's disease, Huntington's disease, and dystonia. And the experimental and hypothetical dynamics in these pathophysiological conditions already shown. We'd like to make our BRA to help to understand these pathophysiological conditions and the therapeutic mechanism of medical interventions, such as deep brain stimulation and lesion therapy. This may also lead us to develop new and effective therapies. So, while making BRA, we have some difficulties. In making. So I would like to provide you topics on how we can make BRA creation public in reality. So here is some considerations on making BRA and on involving other neuroscientists. My difficulties is at first, BRA is a completely new data registration system, unlike writing the paper or something. So we don't have enough time to read through the large amount of manuals. So it's very difficult. And so I think if WBAI like to others to do so need to show a clear future path to produce fruitful results. And hopefully an example already made. And or some incentive, honorarium or other money, so hiring students for making BRA. And more specific questions are these. And while making some connections, registering connection. So the basis of connection is often difficult to register. So pointing single pointer in a published paper is very difficult because this is distributed and sometimes very tedious. And of course, we can use review paper and figure 3 or something, but this is of course, these proxy is can be used, but it is be tedious. And various level of confidence about the basis of connection is there are very various levels of confidence about the basis of connection. So so single axon tracing, maybe some study, in some review. So in some review, area A and area B is connected to neurons in area A is connected with neuron B in area B. But when I deeply seek the papers, these connections are sometimes solely computational one or hypothetical one, so it is difficult to assign the confidence. And maybe another difficulty is assessing the semantic duplication. So semantic duplication is one column. Semantic duplication is one property of the circuit, uniform circuit. But this property is this property means two uniform circuit is the same or different, but there are many names of the same uniform circuit, and very difficult to assess this semantic duplication. And I also reviewed some BIF. So, and on reviewing BIF, I have some considerations. So, one is for ensuring connection, I wonder ensure anatomical or physiological connection or not. So, but I can in this time I can ask the editor Tatsuji-san. So Tatsuji-san clearly clearly answered as it is okay to confirm as the pathway are mentioned in the paper. So if the BIF refers a review paper in review in the description in review paper is can be solely the basis of these connections. So and I think it is useful if the mapping between there is a mapping between circuit ID and nomenclature of the paper. So this is perhaps if the BRA, if some BRA, if in BIF, neuron A, there is neuron A, but nomenclature in the paper of the referred paper, the neuron A is appears as neuron B or C or D, and so maybe creator of BIF have to show the mapping of circuit ID and nomenclature of the paper. And one more thing is negative connections are not reviewed. This is maybe very problematic. So perhaps I think perhaps it is important to assess all-to-all connections, and some is connected, some is not connected. So I think it is good to describe as such. And the related point is threshold of connection is not shown in the reviewed paper, so very difficult to assess. So, and on the basis of my experience in making and reviewing the BRA, so how can we make? I think how we can make BRA creation public. So I have some suggestions. One is to ask neuroscientists, PI or senior postdoc level as a reviewer. So reviewer can consult with editors. So this process can on-the-job train the peer level neuroscientists as a BRA maker or evangelist, and in the future they may come to use and make their own BRA. And next suggestion is to hire young scientists to make VRA, give a grant or monetary incentive for the investigation. So maybe needed for a short period until it become a standard tool like NWB. So NWB also gave some grant to make NWB from the lab data. So it's a good way to make more BRA. And aim to, so I hope aim to become the de facto standard for building hypotheses about the brain functions. So not only for the blueprint of the brain-like software. Is that my statement? Yeah, that's all.
Thank you for your providing topics. And now we would like to discuss about BRA data creation and how to spread these approaches in this community. And first of all, there is many good suggestions and many problem points. Then first of all, I would like to discuss what is the benefit to create BRA data, and it's very important to create BRA data for the neuroscientists and engineers. Actually, it is obvious for the engineering, like human brain software construction. But on the other hand, the neuroscientists wonder what is the benefit. So, how do you think the benefit for the neuroscience?
Yeah, I have some point. Before that point, I'd like to talk about some credibility of the connection distribution. That is some kind of technical problem.
Yeah.
So in WBA now researching about the evaluation of the connection of credibility. That is depend on the of course the description manner of the paper and for what method is used and what kind of paper and many evidence is accumulated and of course there are many evidence for one connection A to B. There are many paper or some data. So we have to merge that kind of maybe the Bayesian inference. So once maybe the very strong evidence is here, so the other weak evidence is not so important. So merging them. So we have to make some kind of Bayesian inference. And other point is merging the before that, we have to merge many kind of evidence. So after we can establish that kind of credibility assessment method, so then the contributor will only register data, so automatically calculate.
Okay. I noticed the Bayesian inference columns in BI sheet, but maybe I think this connection, maybe we can, it is better to describe the strength of the connection.
Strongness means
weak or strong, weak connection or strong connection, not so yeah, two different things.
Strong
credibility and
just as strong,
strength.
different.
But the strength isn't described in strength of some connections, not described in.
There are sometimes kind of neuron size is a kind of
size,
but now not used now.
Okay.
Size means something maybe almost the number of axons. but that is not used because it is hard to estimate many.
But in many studies of past computational studies, there are many gradations of they set many gradation of connection strength. But in when we make BIF, the strength is vanished afterward. So it is very it's a pity.
So I think we need some refinement of the registration process. So if contributor can add some kind of strength, yeah, we have better to add. So maybe the very difficult point is, of course, strength is just a number of the axon. So it is maybe something relative. So how to explain what is strong? Is maybe the next question.
Yes.
But anyway, that's very important.
It's very important to clarify the connections. And in this sense, also suggest the circuit idea, how to nomenclature and say identification of the between ID. So how do you think about that?
So I'd like to know about that question too, because now we are basically based on the DHBA is RMSD we used.
Yeah. But many BIF have sub-circuit or something, or uniform circuit, or such as 5-HT receptor expressing neurons or something. But then, or so neurogliaform cells, but the neurogliaform cells is included in 5-HT R3 neurons. But PubSpin is review paper. Only 5-HT receptor 3 neurons and the connections between 5-HT3 neurons and other neurons. But in BIF, LGF and BIF neurons are connected to other neurons. So the authors maybe describe LGF, maybe identify LGF and VIP neurons with 5-HTR3 neurons. But this mapping is not there are none. I can't find any information in the BIF. In the in BIF, we cannot find.
Also, we contributor can add the information.
Yes. But in the BIF it's okay. But in the paper.
Paper with many papers in it.
So sorry, I don't understand what is your discussion point just now. So
it's a very technical.
How to identify the circuit ID. For example, I registered the circuit on amygdala, and this was notified, but the paper does not suggest my use of this one. So how to identify the circuit ID is very
So of course, for modeling the brain-like AI, we have to identify some part, maybe the MISI architecture, MISI hierarchy. but in neuroscience, there are very complicated area identification.
Area and the cellular elements.
So we have to for implementation engineer, he needs some well-defined component. But neuroscientist world is not that.
The currently the ChatGPT solve everything though.
Yeah, but the point is that if everything is no, but that is not a clear hierarchy. So so we make some module for calculation. That is corresponding to the some part of the neuron groups. But neuron groups is something ambiguous.
Yeah, right. Yeah, especially so I'm some kind of specialist of the MRI studies, and in the human MRI cases, so there's many atlas. So the name is very different from researchers, and especially so different animals. It's very far from the mouse and macaque and humans. So how to correspond us? It's very technically so difficulties. And currently, so I try to focus on the humans MRI cases and the DKT atlas. And then so we don't have I'm focusing on the common muscle study, but we don't have the DKT atlas. So that means that we are collaborate with anatomists in the Ichinohen, in NCMP, and how to correspond that each region and each. So the anatomists decided, and we believe it. This is the country, so what I did it. But actually, so this is very technical difficulty, and this is the research topic in country. And the other people is doing some of the same the MRI data set. For example, the structural MRI and fMRI and diffusion MRI in the human and the mouse cases in the same modalities. And then how to register each of these. This is actually very challenging topic. So that means that this is currently in the macroscopic cases.
But in our AI researchers' view, that is not really true fact. But anyway, we need some kind of hypothesis, some level of approach.
Right.
That is important for, and if we can implement some set of components, so we can try to observe the behavior, and we can feed back to the more good hierarchical segmentation.
Right. But anyway, I think in current situation, so some people decided, and some people also reviewed it. And for example, the publishing the paper cases, so reviewers are seeking on these years, and then discuss with the reviewers and authors. And in this paper, so we decided. So this is continuously doing these work. It's not, it's very sure, I think.
So anyway, we'd like to verify that kind of a progress. So, so to the maybe the next story.
But it is very important because how to decide the circuit ID and how to decide the connectivity. And it is very important because it is a standardization for the data, and so how to establish the standardization. So it is very important point. So how to agree the shared knowledge of the circuit and connectivity and all the engineer and neuroscience, both of them can agree. How should you? What is a good point?
So I have some comment about standardization. So the one of the most successful cases in the standardization in the neuroscience is DICOM. I think very old years ago, 23 years ago, so there's many MRI instruments, Bruker's and Siemens or something, and each of the instrument is a different output. So we get something about how can we sharing. So this is the same 3D data, but actually very different. And some instruments or companies are gathering on the same place and discuss what is the standardized. And then after this, so we can get the same some kind of DICOM format. And after this, so we are some more modification or more easiest way to the nifty data or now the city and there's some kind of different. But anyway, so this is the firstly, so we agreed with the some kind of most important people's company. But the actually, so in this case, for example, MW cases, this is more so complex. So the MW cases are very few companies, and yeah. So but currently, so there's many kind of neuroscience data, and NLP is somewhat strong, but it's not dominant. And especially the other format is actually existing, and which is better, we don't know. Actually, so we believe finally we need to take off. We have so many people providing, but finally, so some of the convergence will appear, and this is based on, for example, the publication numbers or strongest United States or power gaming. But actually, so the format case is the most important, I think. So how many is very, very important.
So that's right.
Yeah. So that means that firstly, so we should be open as possible as we can. Yeah, NLP community is very open style. And so that means that soon, publishing papers, and this NLP cases, many authors is including on here. So that means that many, not only the United States, the Europeans and Japan, and then so we believe that, okay, so we have no conclusion of the formality. Basically, so some people decide, okay, it's okay, mostly people, so we think that. So that means that firstly, we should sharing the many peoples. And this is the mostly important what I think the standardizations.
So I think something making some data structure or style. So as I said, the engineering side is very important. So Taniguchi-san is something implementing the hippocampus system. But maybe it is not easy to very mapping for the VRA data is not so easy. So maybe make some simplification. So how do you think about the implementation like VRA data or we say the more brain right is that how hard is that, do you think?
BIF data, the information flow data is very useful to implementing computation model. But my modeling is very more simplification to proposed model. I want to check more easier brain structure and functional connectivity and hierarchy.
So actually, so I don't know what is the current status of the BI formatting. So Tanaka-san says that there's a very big manuals, and it's very difficult to read like this. And the country. So I just want to ask that. So in the hierarchy, and it's completely fixed, completely so well defined in the manuals by using the BIF. So you can, if you read the completely, so it will be well defined. Roman, how do you think?
I think case by case.
Case by case.
to aim to construct model level. The level of model several level or modular it's dependent.
And in your current cases, in your papers, how do you feel?
In my case, in my opinion, I want to construct computational model. I want to clear the some module level. It is easier to implement.
Oh, okay. Easy.
And in the that implementation, basically start with something Bayesian model or something deep learning model. So that is something today is not using transformer but state space model or something like that. So that is some part is related to the CS3 and related to there or something like that, but it's not always clear and. Of course, engineers make many types of models. Now, model 1, model 2, and the mapping of the plane is different.
Yeah, so why I asked this question so that we are making the NW file. So in the current hackathon, we're making a database. And in this case, we find it, so there is some tools for that NW. So this kind of very useful tool is existing. So that means that it is very easy to making. And then so we can build. But actually, so in the cases, there's a many flexibility on that format. So that means that the country is not so very good. But actually, so for example, in your BIF cases, there's a manual, it's okay, and finally so output the XML. But actually, so you should make. I don't know. So you should, if the good tool, for example, Python tool, and it's very so easy for many people and the students. And so this is also technically very important for the standardizations.
Maybe ITA.
Beautiful.
ITA as logic.
Like actually, so in the case of implementation, like computation model, it's difficult to define the function. So actually, the structure is physically existence. So they can be visible. So how to categorize? So I would like to ask neuroscientists. So what kind of the function in the neuroscience is defined?
Very
deep question. Actually, leads to the engineering function and physiological function. It should be merged.
Yeah.
So how to bridge? So to bridge the functions based on the neuroscience and based on the engineering, actually this definition should be shared. So
okay, I'm not neuroscientist.
It should be defined.
Functions.
So BRA data, to construct the BRA data, we need to define the function.
So for some case, so navigation.
Navigation, yeah.
Input some visual input, something decoder labeling or something. Acceleration data is input to different path and combine and calculate the position of I'm where here. That is, so we need some kind of implementation or something, explanation what should be implemented. So making the model. Without this model, so we cannot understand that we cannot evaluate the probability of that computation model. So we need that kind of semantics.
In many neuroscience review papers or something, you can find could you find these architecture work?
Yeah. So of course, we have that kind of thing.
Yeah.
But that kind of opinion is something separate and
or psychology.
Psychology and in the neuroscience is a different opinion.
Yeah.
So of course that is not bad, but not so many.
Yeah, but I think so. This is very sort of difficulty because, for example, decision making cases. So decision making is we also believe it existing, but actually, so there's a many visual system, and there's a system is completely included. So that means that only the decision making is not so completely solely the correspondent to the neural circuit. So and especially the decision making, there's many other special sometimes of the decision making, and many people is something have hypothesis and propose and what is your the country, so you also read things the papers, you can find some kind of decision making, but maybe so you need a more specific how to which is completely correspondent here and here and here. But it's for me, so it's very difficult. Seems to be decision making to the paper. So this rough estimation may be possible, but it's more special cases. So we need a more understanding of the experiments and in future works.
Anyway, the semantics must be defined depend on the tasks.
Right.
Task define the meaning of semantics. So it is a very important point. And some kind of semantics is very general. For kind of today's Yamauchi-san paper, the prediction error is a very general. But in the making some modeling some complete system, we have to make some position in this room or something. If we need to add some semantics, and we can evaluate that accuracy or something, that kind of thing in performance. Anyway, the granularity and we need both the task dependent and task independent. Both is important.
So actually, there is many programs to make a standardization of VRA data. For example, the circuit VIF level, circuit ID and connectivity, and the functional level, how to define the function or something. So it's very program. So
we think we have better your first question, maybe how to involve the people to this kind of activities.
Yeah. So okay. Actually, we try to move on these topics. How to invite many people to create the VRA data. So actually now the standardization is not enough in the VRA. But so Tanaka Sensei suggests the PI and researchers should review VRA data. What kind of incentive should we suggest?
Yeah, I think the Tanaka Sensei suggests that kind of something training for the middle level researchers.
So I suggest to peer or senior postdoc level as an invite as a reviewer or some.
I didn't think about the reviewers. Reviewers is very important. Your opinion is very important. So I think that we also discussed with Tazis, and the review process is maybe something entrance point.
Yeah.
the construction of contributing the ideas.
So reviewer can consult to the editor freely, but the creator of BIF cannot consult freely editor or because if the editor deeply involved paper concludes the editors as authors. So it's not good.
I think so.
I think the one way is to invite PIs as a review.
So I have actually the three level, but the most important thing is that you should making a good result, good science result based on this kind of format. My format is not making a good science result, but actually, if you have publishing a good papers or very fascinating result, and in here, we are actually using the BIF and seem to be a very related to that good result. So that means that many people is aspiring for focusing on this kind of format. And but actually, this is very far more futures. But and then we should make in the community-based collaboration is very important. And probably the neuroscientist is very important. But actually, your motivation and you have, but actually finally, the neuroscientists want to get a good result. And what is good result? How to read the BIF? You need to make a strategy or story how to make a good result. So neuroscience is more fascinated and making a community.
Good result making the dietary is something top journal.
I don't know, sorry. But at least fascinated to the neuroscientists. And top journal is actually right.
Anyway, we are step by step. We can do nothing in the 10 years ago, but now we can propose submitted to the journal and that is published. So the next
at least we have to know how useful it be. So for neuroscientists, brain-like software is not the primary goal. So but BIF can be used as a hypothesis builder in the neuroscience research, also in the neuroscience research. So I think if you can show the usefulness of this hypothesis builder as a hypothesis builder, so you can involve more scientists.
I think the hypothesis maybe some good hypothesis and various hypotheses. So related to the things now we can use many large language models. So sometimes we are investigating some automatic hypothesis building is our subtopic of now we are researching. So if we can success that kind of approach, so we made many hypotheses and select and make the paper and excellent hypothesis list paper or something like that. So I imagine from your talk. How do you think that?
Related to this and variable comment from the audience. So from Professor Kenji, so to make BIF and others popular and sustainable, it is important to make them interoperable with other database and model description language tools such as these things. How about this point?
The voice is open.
Thank you. Please, can you comment?
Okay, yeah. So now the data-driven model building is becoming common internationally. So I think rather than doing like a separate effort, so we should like collaborate with different projects around the world. And for example, there are many kind of databases already has a lot of information about connection. For example, the one I put in the chat is like a knowledge space of INCF, and then it can search for different data and publication across many different databases, and including eBrains or kind of Dandy Archive. And then they also support some kind of API. So rather than doing everything from scratch, so utilizing those existing resources would be very important. And also in the case of kind of model building, so there are some tools, for example, the Sonata from Allen Institute is one thing, and this is another project called NetPine. So they like hoping to could realize some kind of incorporating data and then produce a network model and then do simulation. So this is more like a single cellular level, like a spiking neural network model, but similar method could be applied to the kind of a more mean-field kind of model. This brain initiative is trying to build. And also the important feature of this WBAI is not just anatomy and physiology, but some kind of like a computational functional hypothesis integrated into the model building. So by kind of outsourcing many functions from existing database and tools, we should focus on how we can integrate the data with the kind of a computational theories and then the functional hypothesis. So yeah, so if the WBAI effort is just isolated, I think we cannot catch up with the progress around the world. So we have to be connected with all the other efforts and then utilize what they offer, and then like recruit people in other communities. That's my comment.
Oh, thank you for comment. And yeah, I think it is an important point, but we are not so resource until now, so we don't have doing this kind of activity. But you suggested the 2 points, knowledge space and NetPine. So I'd like to consider about what kind of association or combination of the kind of activity is better to ask, is that we have to think about next steps.
But I think so. At least we should compare the literature review is very, very important. So what is the difference and what is the advantage and disadvantages? And we need some kind of list. What is the difference for making a document for the introduction or something? And there's a knowledge graph and there's many things. And what is the BIA and what is the difference? And then so we can be how to collaborate with peers, not and especially focus on. So I think so. Yeah, studying so it's a very good point.
Actually, time has coming.
I think the last point is very important. So we should talk about that.
Okay, so last point is last point, advancing understanding of behavior change mechanisms. So integration of the very dynamic and VRA-driven development. So this is a big improvement. And how should I go? Is there any comments?
I think so.
Yeah. The first point is now the building a VRA is not so easy for everybody. So we have better concentrate some VRA that is very connectable with this behavioral change activities. So some behavioral change experiment, and we should build some VRA for focusing the resource. It is I think the next year, this year, next year. I think very important. So.
Yeah, I think so. There's a future, future work, and then more the 1 or 2 years work. So this is very different. I think so. This is a sort of Gakushu Tenkatsu project. So that means that we have only the 2 years. So that means that we should focus on the sort of writing some kind of document, papers or bioarchive is firstly so focus on. And we have already published the bioarchive papers. For this case is a decision making operant conditioning task. We have making so NW file, and this is a readable task. And this is one possible start point to that collaborations. And in this case, in this presentation, so there's I think the two relationships between the first is the second speakers, and this is the decision making and the cortical regions and some of the subcortex. And we have the wide field large imaging of the cortical region. So then this is the one focusing point. And Tanaka-san have already somewhat did it with this breakable task. You also somewhat failed. But starting point is that we have the NLP or big data, and so we have some of the considerations about related to the task. So that will be the starting point of here, and at least 2 years. So we should write down some of the document and submitting the bioarchive or somewhere is I think very important.
Yeah, I completely agree.
Task is shared, so it is important that sharing task is shared.
Yeah, sharing task is very important. And maybe as I think as automation from our NWD to VRA is very helpful. I think.
Using a ChatGPT.
Not just ChatGPT.
What's the meaning of the automation?
Thresholding or something.
Thresholding?
So connection is.
I understand. So this is also a very important point. So using RNC connectivity or something.
Yeah.
So
from our.
For example, so we can make the functional connectivity by using a wireless answer.
Can be calculated and thresholding and automated VRA.
It's really possible.
VIF file.
So that provides VIF.
Yeah.
Right.
So I don't like to.
Sorry.
I don't like to keep that. Excel spreadsheet format. So I like to stream into the data
from NWD data to VIF structures and WBI construct FRG and HCD based on that.
But VIF is possible, I think. So we have the R Institute or the cortical perforations atlas, and we have making time series, so that we can make the correlation between the regions. And the strongest is not. It seems to be it's not completely equal, but actually, so this is a very rough screening is possible.
Any comments?
Oh,
all right.
Come on.
So yeah, I certainly missed most of the discussion here, but so yeah, one thing I heard about it's about the how to accumulate the evidence for individual connectivity, and I presume that there are many types of evidence about inferring the connectivity, and I'm not really sure right now about how BIF or the BRI interface actually handles these different types of evidence.
Yeah, now we created this data.
Yeah.
Now I remember Ashihara-san is making the database. Now we can use LLM. So we gather the many papers and extract the connection data from the papers and making the database is one point. So of course that can cover all things, but we can cover many things from that way. So that is one strategy. Now we are progressing.
So yeah, let's say there are many ways to say there is some connectivity between two regions or two types of cells.
The identification of the region area.
No. So
maybe Bayesian inference or something is a
no. So basically there are so the connectivity can be inferred based on, let's say, for example, the anatomical evidence or electrophysiological evidence or like the transsynaptic traces, those things. And I think this evidence different evidence suggests similar stuffs, but the implications are very different. Implications. So the implications of individual data are very different, and I'm not the fan of mixing everything up to infer one connection, and rather I would like to see the, let's say, kind of building some the decoding or inference model of how likely this cell affects this cell, for example, at the individual at the basis of individual connectivity.
One point is we are going to making the AI model for past purpose. So in that point, we don't focus on the single cell. So to make the this level, so we are some kind of a mesoscopic level. So we can not step into the too detail in that case. But of course, in the neuroscientist view, there are another desire. So it's just a little difference. But in this level, we are not so stepping detail. Is our starting point. But so we define the uniform circuit as Tazusan explained. The uniform circuit means something local same cell type neurons cluster. That is a starting point of computational modeling. So it's our point.
And also, I have actually another point about categorizing the concepts of, so let's think so you talked about how to define the nomenclature of various neuroscience concepts, and I think that Nakai-san mentioned about the DICON or the NWB, but the standardizing the format of methodology is somehow different from standardizing the concepts, like what regions are there in the brain, or what types of cells there are in the brain. So I think that yeah, I guess there may be some other ways of categorizing those things. Like I would suggest that the top-down approach of mapping mapping the concept space. Like so, let's say if you have the whole brain here, and then we can, for example, let's say just locate some vague area of interest and name that region without explicitly dividing the space arbitrarily. So, I mean, at least for the experimental's point of view, that's more or less the reality. I mean, so we just focus on the space based on the stereotaxic coordinates, for example, or based on the shape of the sulcus and gyra. So, so it's more kind of more space-based or partition-based approaches may be more useful for at least for experimenters to annotate the regions of interest or other those conceptual point of view, and also it may be also possible. It may be also amenable for classifications of individuals' neurotypes. So that may be useful, I think.
Related to that point, at least in the modeling, the computational model by engineering side, so we divided some more grain sizes more small, so we can use them dynamically change the combination of this cluster. But if we do not define the minimum cluster, minimum granularity is so we cannot step into the implementation, the smaller than this at that level. So what is most fine grain level is important, but too fine grain is hard to computation. So in the very simple level, in turning some levels, just simply the dated gyrus and CA1, but that is too big area. So at least we have to divide more divide 3 or 5 areas or longitudinal areas size. So that kind of dividing is always important, and we need some more small segmentation is introduced. Need for some functional explanation is our research progress situation.
Okay.
So it is very difficult point to understand or share the maybe concepts of the.
So in his research, some part is defined one unit, but he needs some separation. So functional explanation.
Yes. So function. What kind of function is needed is very hierarchical. Also, it is very difficult. And so maybe more detail can be realized. But how deep should we define the functionality to the minimum size? It is a very big problem. And yes, as Yamakawa-san pointed out, the brain size is very small. It is difficult to define the function. So we should, good point. So like a mesoscopic level or something. Yes. So we have to discuss more good point between the engineering and the neuroscience.
But that is the standard point, so we should do every area, depending on the functional situation of the brain area.
Yeah.
Thank you for your comments.
But anyway, the cortex and cerebellum and these new brain area is something repeated structure. But more old brain area is more specified.
more
intermixed. There are sometimes very simple semantics, but very complex. We say sometimes that new brain area is something like Kyoto city, but other old brain is more like old city.
Like Sahara Desert.
Oh, thank you for the discussing. It's really great talk and discuss, and time is so up and procrastinating. And so I'm sorry for not good time management, but it is very useful discussion. And so 3 discuss points and key challenges in establishing a framework, and challenges in standardization and dismissing open science data, and last topics is advancing understanding of behavioral changes mechanisms and how to collaborate neuroscience and engineering. So we have to more discuss, but today we have great discussion, and thank you for your participation and keep on discussing. Okay, that's all. Okay, thank you for your discussion and participation.
Thank you for attending this workshop. So I have a final announcement. So we will maybe have the next this workshop at the third international workshop, maybe this summer or autumn. If we can accept it, iconic is one choice. And next slide is, so this is Japanese announcement. So これは全農アーキテクチャは今年で10周年になりましてですね。10周年になったので、松尾先生とか栗原さんとかに来てもらって、ノーマタ AGI の実装と活用という、だんだんその実装する方のレベルの方の話をしていければなというふうに思って、一応9月4日の午後ということまでは決まってますということです。ということで、Thank you for inviting us as in the brain behavioral change hackathon, and thank you for everybody attending and making some speech. Thank you for everybody. Thank you.